Hello students. So in this video we'll discuss about meiosis first, next part. So that is, in meiosis first we have discussed in the previous video about prophase first and its subphases, that is leptotin. Yes, the next one is zygotin and pachitin, dikinesis, and diplotin and dikinesis. These are the phases we have discussed. Next one is metaphase first. We have to discuss in this video anaphase first and telophase first. So all these three stages we'll discuss in this video. First, I'll choose the metaphase here. Just after prophase first, there is a metaphase first. So in metaphase first, what are the changes we may observe in the cell division of that meiosis first? See, in this diagram I'll show you. So actually there is no nuclear membrane. Nuclear membrane is disappeared in the previous stage, that is in dikinesis. So it was started dissolved in the diplotin stage and completely disappeared in dikinesis of that subphase of the prophase first, right? No need to show that nuclear membrane, but we may show that esters, right? So here you can show the esters. This is what just I am showing here, the esters, right? So and in the metaphase what happened? So there is a chromosomes, the homologous chromosomes. So they are appears like this. In the equator they are arranged, and the exchange of genetic material where takes place. This is what can show the genetic material here, and this is a homologous chromosome, right? And one more pair of, I have taken 4 chromosomes totally. So here we should show that in the equator region, we can show this chromosome arrangement at the equator region. Right, this is what the diagram we should show like this, because exchange of some portion here due to crossing over, and when the synapses is formed, right? So exchange is done with the uh, endonuclease and ligase enzymes, as you know already. So this is what the thing. Yes, you can also show this one more spindle esters, that is which produces the spindle fibers like this. So we can say that the ester it is, so which forms the estral race, and these are called spindle fibers. So actually here kinetochore you can show, kinetochore, so which is present at this centromere. So spindle fibers are attached to the kinetochore like this from this opposite esters, right? So this is what the spindle fiber attachment. So this phase that represents that metaphase. Yes, because so we may observe that this is the equator region, at this equator region, these homologous chromosomes are come and they are aligned at the center of the cell, that is called equator plate, right? So this is what the metaphase, right? So here actually tetrad become attached to the spindle fibers. So you can point out here some what you call some points you should remember. Tetrads move towards the equator plate. Tetrads move towards the equator. This is the first point you can add. And second point, so here tetrads become attached to the spindle fibers. Tetrads attached to the spindle fiber. Spindle fiber by kinetochores. Spindle fibers, that is actually the kinetochores, we can say, fibers through their kinetochores. What you call kinetochore? Kinetochores means these are the structures present around this what called centromeres, right? Centromeres, right? Centromere. So this is what the centromere, here bracket like structure what we observed around the centromere, that is called kinetochore. So where this to the kinetochore these spindle fibers are attached, right? So next one is, even we observed that the two parallel metaphase plates. So this is what we call two parallel metaphase plate we observed here, because these homologous chromosomes are at one pole and another homologous chromosomes at another pole. So we may observe this, right? So this type of arrangement is said to be, means the chromosomes are arranged at the equator, is known as the metaphase, right? So we go for the next phase, that is the anaphase first, right? So we'll go to the anaphase first now. So next, after metaphase first, we go for the anaphase first, right? The anaphase first shows that somewhat the elongation of cell may observed. Short, right? Yes, here we can observe the constriction, cytoplasmic constriction. Short, cytoplasmic constriction may observed here, and this forms the cell division in cytoplasmic division, actually that's cytokinesis. So here the esters forms the spindle fibers which are attached to the kinetochore, and they are pulling towards the opposite poles. This is what the changes we observed, right? The all chiasmata are disappeared here, actually. You can maintain the points, all chiasmata are disappeared. The all chiasmata are disappeared here. So the homologous chromosomes separate and move towards opposite poles. Here, next point you can add, homologous chromosomes are actually move towards the opposite poles. They get separated. The homologous chromosomes are, this is what the homologous chromosomes which are get separated and move towards opposite poles or towards the esters. So these are the points we should remember in the anaphase as well. This is what ester, and these are the spindle fiber, and these are the chromosomes, right? So these are the changes what we observed here in the anaphase, right? The centromeres do not divide here, that also you should remember. The centromere not divided. One more point you can add, centromere not divided, right? So this is also one of the important point here we should mention. The centromere is not divided. These are the points enough to write, right? And the next we go for the telophase, right? Yes, in telophase the constriction is become more, and the nuclear membrane reappears. Exactly, the chromosomes are moved towards the opposite poles. Homologous chromosomes move towards and reach the opposite poles, and esters are disappears, and nuclear membrane appears here. Nuclear membrane for both set of chromosomes where appears. So means here one set is comes to the one side and another set will come to another side. This is what the haploid set of chromosomes are moved towards the opposite sides and forms a two daughter nuclei. This is what the daughter nuclei are formed here, right? So this is what the changes what we observed here. That is a very important thing. The chromosomes are reached the opposite poles, and a nuclear membrane appears around these chromosomes, and even chromosomes are starts recoiling, means decoiling. So due to decoiling there is a the condensation is released and the chromatin threads are formed here again, right? So this is the telophase, right? And next I'll take here the cytokinesis. So you can observe the cytokinesis. The after cytokinesis there is a formation of complete two cells. The construction is this is what the chromosomes, and the chromosomes may forms again this what you call the chromatin because of decoiling process, right? Like this. So these are the said to be two daughter cells. Daughter cells are formed here, right? The daughter cells are formed after cytokinesis. This process is said to be cytokinesis. We called two daughter cells are formed after just meiosis first, right? These are said to be haploid because only one set of chromosomes we observed here. In the telophase we may observe, yes. So actually the the nuclear membrane is formed around the decondensed chromosomes, and nucleoli reappears, and the spindle fibers and esters are disappears. Spindle fibers also disappeared in the telophase, and the even esters also disappears, right? So next nuclear membrane is appears, and nucleolus is also appears, right? After cytokinesis complete, the constructions reach the center by this way. So there is a formation of two daughter cells by the end of cytokinesis, right? So the next video will discuss about the meiosis too, right? Thank you, students.